So, welcome everyone to the first Recon community meeting of the year. I hope you had a good start to 2025, and for those who celebrate it, happy Chinese New Year, Lunar New Year. So for our first meeting this year, then I want to introduce Aubin Ramon. He is currently a PhD candidate at the University of Cambridge, where his PhD research is on advancing nanobody discovery and engineering with sequence-based machine learning. This is the topic I will be presenting today. He is in the group of Somani at the Center of Misfolding Disease in the Department of Chemistry at the University, where the group focuses on the roles of antibody in biomedical research, as well as in the diagnostic and treatment of many diseases, including cancer and neurodegeneration. Prior to this, he gained his Diplôme Grande École at the prestigious École Normale Supérieure in Paris, where he majored in chemistry with additional biology courses. So alongside his PhD research, Aubin is a scientific consultant in protein engineering using in silico tools for biotech companies like Chimers and Nanoneuron. And recently, he also interned at the Center for Science and Policy. So he really has a range of experiences behind him already. So now we'll just hand it over to Aubin to talk to us more about nanobody discovery then.
Great. Thank you very much for this kind introduction, and Kai. And also, I would like to thank Riklon and Jenny for the invitation. As you said, I'm a fourth year PhD student, so soon to be doctor. I'm almost done. I'm about to submit my thesis in a few weeks. So although I might be a bit sleep deprived, I'm quite excited to be here and to share my work. As I said, it's kind of like a nice way to conclude on my work during my PhD, and to talk about something I'm quite excited about, which are nanobodies. My PhD was mainly computational, so I'm going to take a computational focus into that. But yeah, so let's talk about advancing nanobody discovery engineering with machine learning. So I will mainly give overviews on nanobodies and what are the different available approaches and tools to use to design in silico nanobodies. And if I still have some time, I'm going to talk about my research, which is the thing I know the best, where I developed two computational tools, Native to to assess the antibody nativeness of a given sequence, and Nanomelt to predict the nanobody thermostability. So first, let's start with nanobodies. So if you want to talk about nanobodies, we need to talk about antibodies. So antibodies are these Y-shaped proteins with two sets of two chains. We have one heavy chain paired with a light chain, which is quite important. And more interestingly, they bind to a specific target protein, which antigen, through this variable domain over there. And more specifically, through this complementary determining region, which are named CDRs. Yeah, this is where they're going to bind to the target. So why are we fascinated about antibodies is because, I mean, first of all, part of our immune system, so it's quite important. And they have good developability because they are part of our bodies. And it's one class of protein, one single class, which is able to bind to a wide range of proteins with high specificity and high affinity. And because it's only one class of protein, you can then design like pipelines, libraries, etc., for antibodies that can bind to many different targets. So then, yeah, you can have approaches like libraries approaches where you construct a library when you mutate, for instance, of a variable domain where this is where you bind to a target, and then you can screen it to your antigen and have an antibody ready. And so you have a framework that can be applicable to many, many different applications and many different types of antigens. So that's why it's highly valuable. And even more, you can even like harness like people or animals' immune systems, and they can even like generate antibodies themselves because this is what bodies have. And so you can immunize, for instance, a mouse or whatever, and then it's going to generate antibodies for you, and you can use directly the serum from these animals to make things very handy. So this is antibody. So one more about nanobodies. So first here, let's talk about heavy chain only antibodies that can be found in camelids. So it's a variant of antibodies that only have a heavy chain, so we don't have a light chain. And this type of proteins are quite interesting because this is a perfect example of serendipity in science. So in the early 90s, we have this professor in Belgium that was giving this practical course where they had to sequence antibodies And they give us, as bloodstream, they give like a serum from camelids because this is what they had. And then one of the students came up and said like, oh, it's funny because I found this very small version of antibodies because they are smaller. Here we talk about 80 kilodalton compared to 150. And then they investigated and they found that camelids naturally produce these heavy chain only antibodies, whereas they also produce the antibodies, but we don't, as humans, produce this heavy chain only format. And then if you take only this variable domain over there, which we name VHH, we have a single domain only antibody that targets through the CDRs an antigen. And we call it, this is what we call nanobody, because they are in the nanobody range. The size is a few nanometers. And this is the structure of a typical nanobody. It's just a beta sheet fold, which has these 3 CDRs, so the loops are going to bind to the target over there. And this is what I'm going to talk about, and this is what I'm excited about. So why engineering nanobodies? How do we care about them? I mean, what we see directly is that they are smaller, so they can be useful. But if they are smaller, like we can use like this single chain fragment. So we can generally take the fragment of the antibody, and it's quite small too. We don't need necessarily of this concept part. So this is what people being engineering as well. But the main aspect of nanobodies, they are single domain antibodies, but they only have one domain. And because they only have one domain, they don't have this hydrophobic patch that you need if you want to pair these 2 domains together. And so because of that, that makes them less prone to aggregation. They can also more easily like refold, and so they are more tolerant to thermal denaturation or pH denaturation dependent. And because it's also only one domain, it's much more easier to express, and we don't have to care about this pairing between these two chain, but is needed to bind to the target. So if you don't have a very like precise control into this interface and this pairing between these 2 domains, you're gonna struggle a lot. And this is what people have been struggling a lot. Sometimes just the pairing of this heavy and light chain is difficult to engineer and handle. Whereas here we don't have this. With only one domain, you're able to bind with affinity similar to full-length antibodies to your target. They are smaller, so better tissue penetration. It can even be used for nasal administration, things that can't do antibodies. It can even be used as intrabody, so in intracellular environment, because they are more tolerant to redox condition of intracellular. And then can also cross more easily the blood-brain barrier, which is very difficult with antibodies. And they also have a longer CDRs, free, just by definition, this is how they are. And because of that, also because they are smaller, they can have a better access to cryptic epitopes and to, better epitope accessibility. And so you have to stretch my reasoning. So this is not exhaustive, obviously. And some of those properties don't have necessarily a clear consensus. Some people might say the opposite as well. But this is, basically why we like nanobodies. And if I stretch my reasoning a bit more, because I have to say what I'm doing, this is the best of like both small molecule and protein therapeutics worlds. Protein therapeutics because it's the protein. We can have like all these protein engineering pipelines, use them. They can bind to a variety with high specificity and high affinity to a variety of proteins, but they're also like smaller, so better tissue penetration, etc. So this is why we want to engineer antibodies. Then what people have been doing with these single domain antibodies, then you can create like a zoo of formats and constructs where you can like bind multiple like VHS together to be like multi-specific in different formats to be available in bloodstream or for different applications. But one application I like to highlight, which is the one I think is giving more and more attention, is to use nanobodies in CAR T-cell therapies. So this is nanobodies-based CAR T-cell therapies. In traditional CAR T-cells, they use in the CAR T-cells, they're going to use single chain fragments like those ones. But because it's more complicated, etc., and more difficult to have like a nice pairing and have stability and to be stable enough in a tumor and cancer environment, people tend now wants to use nanobodies. So they've been engineering this nanobody-based CAR T-cells. So I think it's one of the applications pretty cool. The only question we might have, and the question I had, is like, why don't we have nanobodies in our bodies? Why did evolution didn't give it to us? So we are not sure about like what's the history of evolution and why camelids have it. Because camelids have nanobodies, but also sharks, quite interestingly. They are slightly different. I think more attention is given right now to camelids, also because if you want to humanize camelids, it's much more easier because you can have camelids farms, especially in South America. They have a lot of them there, but also in Europe. Whereas like shark is a bit more difficult to humanize them. Even like some people have been trying to use in China, I've read shark farms to generate repertoires of nanobodies. But interestingly, just two different events where while one of the hypotheses is that there was like this epidemic, this kind of virus or bacteria, whatever, that necessitated antibodies with high stability in for instance, like with maybe a very specific pH, or I think that was the hypothesis for the shark ones. And nanobodies were there by mutation, they got selected, and then the population of camelids or shark that had these nanobodies got selected, and then they had it. So this is one of the hypotheses, but we're not still sure about it. But nature didn't give it to us. So are nanobodies that great? Do they have some limitation? So one of the limitations people might think about is immunogenicity, because if we don't have them in our bodies, if I put a single domain like that one, our immune system might react because it has never seen a single domain antibodies. It's used to seeing a heavy domain paired to a light domain. But here we have like a new interface, hydrophilic interface here that the immune system haven't seen. So it might generate immunogenic response. So that's one of the reasons. Another reason is that possibility is that because the CDR3 is much longer for nanobodies, but might generate new conformations, new set of peptides that the immunogenic system might react to. We don't have a lot of data about this immunogenic response because it's pretty new class of proteins, especially that the patent of nanobodies got uplifted, stopped, whatever, like 10 years ago. So it's only since like the second part of the 2010s, etc., that we started to focus a lot on nanobodies. And the first approved drug was approved in 2019, which is clobemicimab as a nanobody. When now I think we have two more, at least there's one in China, in the Chinese market, has been approved. And for these two, the immunogenic data we had from clinical trials show that they still have some immunogenic response, even though they've been humanized beforehand. So we don't have clear consensus yet. We don't have a lot of data, even like cellular models, and haven't been developed too much into nanobodies. But I think the main consensus is like people just tend to humanize them in any cases. And then another like drawback of nanobodies is like, okay, they are smaller. So as I said, they better tissue penetration, but they are too small. But maybe sometimes the clearing can be too fast because they can cross the renal filtration sieve very easily, which can be quite suitable for different applications, just like tumor imaging. But this drawback is actually easily like fixable if you want to increase the half-life time. You can mitigate it by linking it to FC region, so you put back the conservative region, or like serum albumin moieties, etc. And because it's only one domain, they have a smaller paratope diversity because they only have like 3 CDRs compared to 6 for the antibodies. So the diversity is a bit smaller. Because of that, the CDR3 is longer. CDR1 tend to be a bit more diverse than it is for human. And also we do see more often binding happening between the CDRs loop, but also with the framework, because this is accessible. And that might complicate things when you want to engineer nanobodies, because what people tend to do right now, if it's just focus on the CDRs, because this is the determining region that's going to bind. But now we also have, with nanobodies, we have to be aware that binding can happen within the framework as well. So you need to be careful with that. So that's our nanobodies. Where to find them? So libraries. So either you take a llama, you take his immune repertoire, native one, or either you just humanize it with your antigen target for llama, or you can even like construct like synthetic libraries. So you just take, you just like randomly mutate, for instance, the CDRs or a bit of a framework. Then you build your library, and then these 3 libraries can be used to screen and different displays to your target antigen, and then select the antigen, the antibody, sorry, that's going to bind to your target. This is what people do experimentally. I don't do that because I'm computational. So Oh yeah, sorry, I also forgot. This is very cute. Now people have been developing recently what they call llama mice, which is these transgenics mice that are able to to express naturally, naturally in their body, heavy chain only antibodies, so nanobodies basically. It doesn't look like it. I just couldn't resist, so I used ChatGPT to generate the figure. But I think yeah, it's pretty cool. So these are the different experimental methods to find and generate nanobodies. These are, I think, a quite exhaustive list of available synthetic libraries of nanobodies. The thing here is like for most of the papers, they just actually describe, when I say available, just like people that describe their libraries in the different papers, most of them are not accessible and open source. So yeah, because it's recurrent community and we are interested into open source, etc. So I'm gonna try to make some comments about what's available, accessible. And for synthetic libraries, not always the case. I know for some of these papers, you had to contact directly the research group and collaborate with them if you want to have access to the library, etc. But this is not my expertise area, so I'm not completely updated about that. Why I know more is what's accessible online if you want to do computational. So where do we find nanobodies online? So here we need to give like a big shout out to the work of Charlotte Dean in the Department of Statistics in the Oxford University, where she's been doing a lot of work, open source work on antibody and antibody development. And she's been building this amazing repository online of sequence antibodies from different immune repertoires that she called OAS, so Observe Antibody Space. In that, you can have access to more than 2 billion antibody sequences. And it's built in such a way that it's going to just like automatically update the data set based on what people publish in the literature. So it's quite great. So now, and it's been growing like crazy. Now we have like 2 billion sequences, which is huge. Here, so I focus more on nanobodies. So what about nanobodies more specifically? So they've been building a sub data set where they come like PLAB-DAB nano, which focuses not on repertoire sequences, but on any nanobody that's been published in the literature. So most of them are actually like from synthetic libraries, or they've been highly optimized, so they are not native. That's why it's not repertoires, but it still gave like around 5,000 sequences. And then there's AmNative, which is a data set I've been building, but collates different various sources together from different immune repertoires of camelids. So here we're talking about naive or immunized repertoires of camelids. And altogether, I managed to gather 21 million sequences, and then I've been able to learn from it. And this is open source, the data set. And then when online, so what about protein? We care about the sequence, but also about the structure, because at the end of the day, the structure is what it's going to do, and it's going to act in the binding and in the different properties. And structure-wise, so we have a PDB, and again, so we have Charlotte Dean, who developed this, basically this subset of PDB that can download of the PDB related to nanobodies, antibodies together, and she managed to compile around 9,000 structure of antibodies. This is how we have so far. And then for nanobodies, we have around 2,000 structures. So nanobodies, I mean, bear in mind, it's quite a new class of proteins. So we don't have a lot of data. And that's quite important, because then when we try to predict and learn from a small amount of data, it's a bit more difficult. But still, like the community manages to do a quite impressive amount of work. So now a bit more time to spend on the computational engineering of nanobodies. How do people do what people do? So first, about when you want to engineer protein on your computer, is in the area of protein engineering design computationally, and which is quite a trendy area, because they just won the Nobel Prize this year with AlphaFold and the David Baker Lab, who's been using computational tools. So what is available? So I mean, the very famous one is AlphaFold, which is doing like structure prediction. With AlphaFold 2, that's been released in 2020, which is one that gave a lot of highlight, because they reach like crazy accuracy in structure prediction, and this is why they got the Nobel Prize. And then another area of protein engineering using silico tools is sequence representation, because if you have a sequence, it's just a string of characters, text, if you may, like, if you will, like the residue sequence. But then there are a lot of tools that try to represent in a very informative and nice way that can be used in a lot of deep learning and machine learning models, protein sequences. And so we have this great tool named evolutionary scale modeling. ESM has been developed by Facebook, by Meta, Facebook at the time, which is a large language model like ChatGPT, but trained on sequences of proteins. So this is just proteins right now. And this tool has been useful for so many downstream tasks, sorry. And what about them? Are they open source? So ESM is fully open source, the weights, etc. Maybe sometimes not commercially for the latest models. And then AlphaFold was the case actually for AlphaFold 2. AlphaFold 2 was fully open source, so the code, the weights, the reasoning, everything. And then now we see a trend which is not exactly the one they started with DeepMind, where AlphaFold 3 was released, I think, like half a year ago, and they only made it open source this month or last month. It took it some time for them to make it open source. Now we have the code and the weights only for academic use, no commercial use, because AlphaFold 2, which was great with it was also commercial, open source, so that gave her like so much interest. AlphaFold 3 is not, and I don't know what happened next, if they're gonna be less and less inclined to be open source. But we have other research groups that do amazing people that try to build open source for everyone, such as this community. And we have like Rosetta Fold, for instance, that try also to do like protein structure prediction. So this is the David Baker lab. I also got the Nobel Prize this year. With pretty good accuracy and open source. And then more recently, we have like this model named Boats1. I just discovered it, which is about, I like that model because they call it like democratizing biomolecular interaction modeling or structure prediction. I think it's quite important here because even if like the code and the weights are open source, then the idea is like, if you build like a huge deep learning model, but requires so many GPU to be trained on, but also even like to be used for interference or to predict a structure, for instance. Not a lot of labs have access to a GPU or to like big computers. And so I think the idea now is to build like models that are maybe slightly less accurate, but at least they are fully open source, but then like easily runnable on your computer and everywhere. I think this is where the open source community in structure prediction is trying to go, is like, okay, step one, obviously like open source weights, open source code, but then like building something that is usable by everyone. But you don't need to have like the latest computer and all of that. So yes, I think this is the take here. So what about antibodies and nanobodies? So here with antibodies, which is nice, is because, as I said, is one class of protein that can bind to a lot of targets and generate libraries, etc. So you still generate like a big amount of data for a self-consistent protein, which is this Y-shaped protein. So thanks to that, you can then fine-tune these general protein models onto antibodies to make it more accurate. Because the issue with AlphaFold and this structure, this general protein structure predictors, is that are not super accurate for antibodies, especially at the CDR region, so at the loops that's going to bind to the target, which is like the most important part in the binding. So what people are trying to do is to develop these fine-tuned models on antibodies to be more accurate, leveraging these kind of structures. And plus, these people are great because they're making even more open source than before. So we have like, for structure predictors, we have the Charlotte Dean lab in Oxford University that's been building this immune builder suite where they can generate antibodies and nanobodies trained separately on 2 different data sets of structures. And then we have nanobody, which is named Nanobody Builder 2, that can more accurately predict the structure of nanobodies, even though I think today we are more accurate on antibodies because we have more data. And it's easier to predict antibodies because the CDRs are a bit like, they are smaller. For nanobodies, the CDR3 is much longer and we have less data. So then it's make it more challenging to predict its structure. So we are not quite there yet about predicting with very high accuracy, high enough for prediction, for binding prediction, structure of nanobodies. But we're going there. And then about sequence representation, again, you can fine-tune the protein models into data set of sequences like that paper that did it on nanobodies named Nanobird. But yeah, but that's it for nanobodies. using all these different models, what do you want to predict? What do you want to use it? So there are different properties that obviously you care about and you want to predict when you develop antibodies, either even to be used as a therapeutics or just to be used as an analytical tool. So you want to make sure that it has good solubility. So what people use in antibody engineering is a CAMSO tool that has been developed actually by my supervisor. This has been developed on protein, so to predict the solubility of protein, but this has also been validated on antibodies. So people, our lab, for instance, we use a lot of CAMSO to predict the solubility of our design sequences. Then there's stability. So you want to make sure they have good stability. So there's a tool I developed named Nanomer that I'm going to present. Another tool I focus more on the pH stability of antibodies. Then what we care is about immunogenicity if I want to use them as therapeutics. So there's a tool I developed named AbNative. I'm going to come back on it as well. And then another one named UMatch. This is possible. These are different options. And then this is a goal aim we want to predict is binding. Eventually, what we want is binding. We want to make sure that, okay, good stability, good developability, these different properties. But ultimately, you want to use them to bind to something. So you want to predict binding. Spoiler alert, we can't accurately predict binding because if we could, you would have seen like many drugs everywhere in the market. But there are tools that are trying to do so, that are going there. We have something named antipacity that basically represent a multimer between the antibody and the antigen as an elastic network of a protein. And from that, they train a deep learning network that can show some correlation with binding. But the issue is that you need the structure of your antibody, for instance, that you don't have if you do it in silico. So you need an accurate prediction of your structure, and we're not super accurate right now. So that's why, I mean, at some point we need everything to be super accurate if everything needs to work together. And then we have paragraph that try to predict the paratop residues in the antibody. So which antibodies are binding to your antigen. That can be super useful when you try to do reepitoping, or if you want to resurface your antigen, your antibody, etc. So these are the different tools available, I think, for antibodies and nanobody engineering using in silico methods. What's next? Oh yeah, and what people have been also trying to do is to generate in silico. So this is nice, for instance, if you have like already a sequence, but maybe targets and you want to optimize it, or you want to classify different sequences together, like more like assessment tools, if you will, to discriminate different sequences together. And then you select the best one in vitro for in vitro validation. But what people have been trying to do, and this is a very exciting area of research right now, is to generate in silico a complete whole new sequence. So this is like a very cool video here of RF diffusion. So it's a Rosetta 4 diffusion based model that the Baker Lab has been developing. But tries basically like with diffusion models. So diffusion is just put like a cloud of residues. That doesn't make any sense online, but it's trained to then like to diffuse back the residues to bind to your new environment that you just given. So if you just give a new antigen and you start with a cloud nearby, then it's gonna like learn to project these residues towards the antigen and develop and design a set of residues, so peptide that's gonna bind to it. And what they've been trying to do is that if you give different templates, you can bias the generation in such a way that it's gonna design a specific type of protein. So if you give, for instance, like an antibody template, it's gonna try to diffuse these residues into an antibody-like protein to target your antigen. And so this is a recent work we've been doing, which is still in my archive, not fully published yet. But basically, they take a cloud of residues, a template of an antibody, and given an antigen is going to generate a complementary antibody to target this antigen. The success rate of such a method is 10%, at least, for easy cases, I guess. So we see that there's room for improvement. So I think they've tasted like hundreds of variants and only like a few worked. So which is, we haven't solved the issue yet, but a lot of improvement been made. When I started my PhD, this wasn't possible at all. So here we are. So what time is it? That's already the time. So about my research, like just in a few minutes, that's good. Where do I play a part? So when we do antibody engineering in silico, what happens is like you have a target, which is the antigen. You give it to a computer that's going to generate a new sequence, and then you have like a de novo antibody. So a new sequence antibody, but you're not sure it's going to work in vitro because you are still in your computer. And another pathway is like you have a target, and then you can do screening libraries, synthetic libraries, and have like potential good binders, like a list of 100. And maybe you want to make sure to select the best ones to keep going into the pipeline of developability, et cetera, to generate your antibody. And this is where I play a part, is like, I'm in the assessment part, so I don't do this generation part of the protein engineering here. I have a sequence that is already existing. And then my aim is to predict like good solubility, stability, properties, developability properties to make sure that these antibodies will be relevant for the different applications. So I developed two tools, which is Native, where I basically learn a language model on a data set of antibodies. So human antibodies, for instance, or chameleon nanobodies that are coming for immune repertoires. And I try to learn what does make a sequence of residues, a human antibody sequence, or what does make a sequence of residues, a nanobody camelid sequence, in such a way that then when you give me a new sequence, I can tell you, oh, that looks like actually like a real antibody that you can find in the human immune repertoire. So it's more likely to bind, to fold, to be stable, to have other good property, to be native to a human repertoire. So this is what I mean by nativeness. I try to assess it, and I've been using this method to humanize mouse sequences or nanobody sequences to reduce their immunogenicity. And then I also built another model named Nanomel to predict the thermostability of nanobodies. And by doing so, I generated a new data set open source of thermostability data points. Yeah, I'm gonna skip all of that. Acknowledgements. I want to thank my group, obviously, Peter Somani Lab, for building all these different tools. And I would like to thank you for your attention.
Great. Thank you, Aubin, for the overview about nanobodies, antibodies, and your research. I mean, if people are happy, we can also spend a bit more time if there are more slides that you wanted to discuss about specific research. But should we take a quick pause here if people have any questions before moving on? What do you think?
I can share again after.
Sure. Eric.
So this was really cool. And I know you said towards the end there that you are looking at them for thermal stability. This is a somewhat naive question on my part. I'm not an expert in this field, but is there also any way to kind of think about like specificity compared to other targets within, you know, humans, basically? I mean, you're developing these things on a specific antigen, and then picking out some candidates, as I understand it. But then I'm just wondering if you can then run that through other potential or similar targets to see whether or not, I mean, because this is an issue, if you're thinking really downstream towards like the clinical stuff, that's the one that's going to probably come up the most. So I'm just wondering if that's part of your model.
So it's not part of my tools because they basically are target independent, because I just care about thermostability. So thermostability of the nanobody on its own, or the immunogenicity on the nanobody on its own. There are other people in my group that are working into trying to predict this specificity. But also, as you said, it's quite interesting, is like, okay, so if you have this generative pipeline that just generated a nanobody for that target, what happens if I give it to another target? So what we might maybe want to do is maybe you take the proteome or whatever of human, and if you're fast enough, you can maybe like compute your design and see if you're gonna, of the hypotenuse, if you're gonna have an antibody, a nanobody similar to it. Or what people do, like for more specific projects, sometimes they try to develop, especially if you think about like membrane proteins, target, etc. You want to be highly specific to a subtype of your target protein. So you want to target the alpha and not the beta, and they only differ from like a few residues. So in that case, like when you design your pipeline, the whole point is to be highly specific to that one. So you're gonna design your pipeline in such a way you want, in contrast, not bind to the other. And so that's gonna be part of your own pipeline. So this is like for a very specific project.
Thank you.
Replies to your question. Any other questions? Otherwise, I can just present a little bit more.
There is 2 questions in chat. I don't know if Adrian, you want to speak or if you want me to read out.
Oh no, that's okay. Yeah, we can do that. So obviously, having just one domain would that not reduce the affinity significantly? And also, if you have only 3 CDRs compared to the normal 6, that sounds like you're going to be less specific in binding, right? Can you
So
elaborate a little bit on that? Yeah.
yeah, so what has been found is that the affinity and specificity of nanobodies are comparable to the one of full-length antibodies. So they do manage very high affinity, and they still manage to get specific. So what nature tried to do to compensate the fact that you only have one domain compared to having 6 CDRs in 2 domains is to have a longer CDR3. So then you are potentially more diverse and you can bind to a wider range of proteins. Also, the CDR1 is more diverse actually in practice compared to the full-length one. In human antibodies, the CDR1 is a bit more conserved than it is for camelids. And then you're going to have like a complementary binding with some of the framework residues, what you barely never observe in antibodies. So with nanobodies, sometimes the target is going to bind also with the framework. So that might explain maybe why they still have like very high affinity and they can manage to still be highly specific.
Okay, thank you. And another short question, and this is kind of a general one. How do you compare this one to the nanobodies to the aptamers? Kind of the same idea, right?
Sorry, what do you mean?
The aptamers.
What's
So-
aptamers? Sorry,
Oh, aptamers are basically exactly like nanobodies, but usually are made of DNA and RNA. They are smaller and they have exactly the same. It's a fairly big field right now. Yes.
Okay, to be honest, I've never came across aptamers during my work, so I can't comment on those ones.
Okay, thank you.
I just know that there are indeed like a lot of derived proteins that are very similar to nanobodies. They're called monobodies as well, which is this alpha helix bundle, and they have this loop trying to bind to other things. But I never came across aptamers.
Thank you.
Ooh, it looks like Eric's also shared in chat Adjean's little primer about aptamers, so maybe that's something to have a quick look into.
Yeah,
Might be a question that comes up at the Vibe at some point.
Yeah, I remember during your talk that there was a lot of very nice available posts.
Yeah, questions are coming in thick and fast as well. So Joseph, would you like me to read it out?
I could try if you can hear my voice.
Yes, we can hear you.
Okay, hi everyone.
Hi.
I will try to ask it. Did you try to experiment in silico of the affinity of nanobodies on DNA or short oligos or only on proteins?
So all our pipelines are mainly built to target proteins, peptides. The thing is, our field and our group is pretty new. And so all our work is pretty fundamental yet. And so we haven't like been able to apply a lot of our tools in different projects and different targets. So soon, I guess we're gonna be more involved into the end of the pipeline, which is like, okay, now we can be involved in cool projects and target different things. But so far, we are still building tools, so it's quite fundamental. And one day, hopefully, everything's gonna come up together, and we're gonna have a very robust pipeline to design nanobodies. But so far, it's true that my expertise is still super fundamental and not very applicable yet. But we work on proteins, targeting proteins. Yeah.
That's fair enough. And I'll move on to Brooke. Would you like me to read out your question, or you're happy to chip in?
Yeah, I can. Hello, everyone.
Hello.
Thanks, Aubin, for the nice presentation. Your research is mostly focused on the treatment aspect from the examples I see. Can this be applied for the diagnostics or affinity chromatography method development? Can this research contribute to this aspect? And the other question is that that one maybe a typo in one of the slides. You said what to predict. Did you want to say COMSOL, or is that COMSOL is another device, another computational software, or the popular COMSOL?
So to reply on the second question, it's CAMSOL. CAM is for Cambridge, and SOL is for solubility. And it's been a tool that's been built by my supervisor to predict solubility of any proteins. So it combines like a lot of different parameters to do so. But it's been also validated on antibodies. So in practice, we use it to predict the solubility of antibodies. We don't need to have a nanobody antibody specific one. And the first question, which was, sorry, I have
on the diagnostic, we do have. I am a researcher in Ethiopia. We are trying to develop some aflatoxin quantification methods, and for that there is a research. There is a research on developing a monoclonal antibody so that you can concentrate, you can trap these aflatoxins, and then to quantify this low-cost quantification method to develop the presence of aflatoxin. So how can the implication of your research in developing such kinds of quantification methods?
Yes. So that's true. My main spec was more about therapeutics. So using in bodies, which makes it a bit more complicated, because then you care about immunogenicity, be specific a bit more. So for instance, like one of my tools is to predict stability, and that is important even like for shelf life, shelf life of your antibodies. And just in general, the pipelines just take a target, and then it's gonna design a complementary antibody to bind to it. And this can be done for targets that are going to be used for either for diagnosis or just for quantification methods, as you said. So they're completely applicable to those purposes. And actually, a few projects we do here are involved in diagnosis, for instance. So to build this, how do you call it? Antibody, during COVID, we're keeping using it. This antibody test, flow lateral test. Yeah. So for example, we've been developing those ones as well. But for that, you still need like a good solubility and for the buffer you're going to use, and good stability as well, etc.
Thank you. Thank you.
Thank you. Now call on Jenny. You had a question for Aubin?
Yeah, I guess it relates to the question earlier about the specificity, because we've got a project currently with an MPhil student where we're trying to make hot start antibodies for a PCR for DNA polymerases. So these are antibodies that basically bind onto the DNA polymerase. And then as the reaction gets heated up, they disassociate, and the polymerase, which is thermostable anyway, is free to do its thing. And this is helpful in a diagnostic context, because you basically you avoid non-specific amplification if the DNA polymerase is only functional at a higher temperature. But most of these sequences are proprietary, or they're now aptamers. A lot of them are aptamers, but they're not published. So we're trying to, but in that case, we were basically trying to find stuff that isn't totally specific, because we kind of want it to bind to different members of a protein family. And I sincerely apologize, because I had a clashing commitment. I'm not going to watch the recording to catch up on the talk. So maybe you touched on this already, but like, are there ways in which you can leverage the ability of antibodies to be nanobodies, even to be less specific, and to potentially kind of have these types of multiple binding targets within a protein family?
So it's funny because usually what we try to do is to be specific.
Exactly.
Well, I think like the easy answer would be to say like either like you do constructs with multiple fragments,
Mhm.
but because like Nanobody is just a single domain, it's easy. Then it's quite easy to put like a lot together with different linkers. So people have been trying to do it. If you want to have, I mean, it doesn't reply to your task, but if
No,
you
I
want,
think it does though. Yeah,
but
so
like,
be
like
specific, but put them together as a kind of multivalent
Yeah.
nanobody kind of structure. Yeah, and that's one approach that the MPhil student is exploring in the project
Okay.
because we're not sure. Yeah, we're not sure if we can be like specific but not specific
Yeah, I mean,
enough.
it's like a sweet spot that you want to reach. Because if you're not specific enough at all, then you're just gonna buy anything, just like protein A or whatever.
Yeah.
So, I mean, we can use potentially like an approach, as I mentioned earlier, that can be, I know you can screen to your different targets, because I guess you still know what you want to bind or family you want to bind. And maybe you can try to find designs that are optimal for all of those families as well. And
Yeah.
this is very useful for within silico design once we get there, is because it's quite fast, and then you can iterate to the different targets you want, and maybe you can find like the sweet spot that are gonna bind to everything.
Yeah. In our case, we're looking at single chain antibodies because we don't have access to a nanobody library, but we do have access to a very large single chain library.
Okay.
So they've got the epitopes are now synthesized and they're doing the screening process. So we'll find out how many we can add together. But then, and single chain, you can also, if we end up having to stick multiple together, I mean, you can do single chain as well, but nanobodies are even more kind of amenable to this approach. So yeah.
It's more easier to engineer multiple nanobody domain rather than multiple single chain one.
Absolutely. And just a note for everybody, of course, once we hopefully have identified successful hot start antibodies, we will be publishing them all to the Reclone forum and the collections so that you too can run hot start PCRs. I mean, part of the problem is we have some of the polymerases in our open DNA collections. There are no nobody really uses them as hot start. so even if people did publish in a patent, say the antibody sequence or the aptamer sequence, it doesn't exist for some of these. And so yeah, we're hoping we can fix that. And Eric, they'll be in AdG, of course, just if they function. Like a big question mark to begin with for the project, but
Yes, definitely. And just to follow up on the specificity question, Adrian, he was asking about any tools for predicting off-target for generated nanobodies, if that makes sense. So I think the question was directed to you, Aubin, and Adrian was just asking if there are tools for predicting off-targets for generated nanobodies.
So for nanobodies, no. For antibodies, I think there are some attempts, but it's not clear to me in minds. There's a project going on in my group that tries to do that. So I guess there's a lack in the literature. But no. I mean, there are, like, as I said, ways to see if you are specific, but no directly. I mean, directly predicting this specificity is a bit hard to just given your sequence and your antibody, your protein is a bit hard. You need the knowledge of the whole proteome, the knowledge of what's your diversity that you can reach with your nanobody and antibody, I guess. But I mean, I'm just making up an answer right now, but I need to look it up a bit more.
Not a problem. Thank you, Aubin. I think, Joseph, you had a follow-on question from earlier. So he says, I think FC could be appropriate more than nanobodies. Is that right? I assume that's flow cytometry there? Or
It's a comment for something earlier. I think the crystallized fragment is used, I think, for a lot of proteins, like human proteins or camel proteins or rabbit proteins as a general. So they could be for a broad range for polymerases also, if someone could design them. It was just a suggestion and it's more of a question.
But yeah, antibodies are used with framework constructs sometimes, and most of the time actually, for better half-life time in the body, if I may add.
And there's an additional comment from Eric, just to say that there are tons of tools available for off-target CRISPR. So I was wondering if there's something that can be adapted here for nanobodies.
No, I had a look. And add a section in my thesis about this introduction. I still have 1 week.
Very tight deadline. Very glad you were able to join us today. I know we are
Yeah.
running short on time. I'm happy to stick on for a bit more if folks are interested in hearing a little bit more about the other slides that Aubin was presenting. I don't know if you also have time, Aubin, or
I
we
have some
can.
time. But
Yeah, so I'm happy to hold on for another 10 minutes past the hour if you'd like to continue presenting, if there's no more questions.
Yes.
And if you do have to leave, feel free to drop off, obviously. And we will share the rest of the recording. So if you do miss something, you can always come back.
I am ready. Yes. So 2 tools. One to predict immunogenicity, nativeness, and then thermostability. So as I mentioned so why do you care about nativeness? So humanness, obviously, to have like lower immunogenicity, and you want to design a sequence that looks like human one. So they have like all the good pharmacokinetics and developability properties to work in a human body. And then for nanobodies, so we don't call it camelidness, we call it nanobody nativeness. And you want to have like a sequence that looks like the one in the camelid immune repertoires. So the one that are all the good properties I described in the first part of the talk. So good stability, they can refold, like environmental refold, etc., etc. Sorry. I think maybe the room is booked for another meeting after. I keep them for a few minutes. Then that's it. So what it does is it uses this machine learning model, which is like this bottleneck over there, that takes human antibody sequences, repertoires, and it's going to try to learn the most efficient representation in this bottleneck. So the most meaningful information that is shared across the human sequences. And so that's going to learn what makes a string of residues a human antibody sequence. But doing so, if I give like a human antibody, it's used to see it, so it's gonna like reconstruct it well. And when I give a mouse, it's a bit more lost, so it's gonna project the sequence a bit further away, and the reconstruction is gonna be like worse. And if I give like a very random sequence set of residues, it's gonna be completely lost. And I'm gonna use this reconstruction task to quantify and assess the humanness. So then if I give like a data set of human sequences, I'm going to give like a score pretty high over there. If I give mouse one, I'm going to give a score lower. And so this is just to scare people off. But what I've done is I created like a rescanning in such a way that then upon 8 becomes like an interpretable threshold that people can use when they read the score of the sequence, saying like, okay, that looks human because it's above upon 8. If it's like upon 4, that's not human at all. Then how can we use such assessments? We can score data set, as I said. We can have a profile because this score can be residue specific. And so we can see like when there is a peak, it means that this is not considered human. So here we have a mouse sequence, white type, there in yellow, with a lot of peaks, it means like a lot of like non-human residues. And then it has been experimentally humanized, like when they develop that therapeutic. And then we can see that the mutation suggested increase humanness over there. And then that humanness can be correlated with immunogenicity, as we saw some clinical trials. So this data is a bit not, because between the different clinical trials, they do it in different conditions, different injection, different concentration, different pools of people, etc. Sizes pools, and this data isn't very consistent. So we see some trend between like the idea of mouse sequences, therapeutics, and the one of human one. But we don't have like a perfect correlation. But this is what people report in the literature. And then what I did, which is quite an interesting part, is that I tried to combine the nanobody nativeness with humanness to humanize nanobodies. So we have like a cute scheme here where I take nanobody sequences from camelids, I put it in my architecture, and then at the end I have more human-like antibodies. So what we tried to do, we did apply it on 2 nanobodies, where we took the white type with like its humaneness score and its nanobody nativeness score here. And then we suggested mutation through different pipelines and strategies in order to increase this humanness while controlling for the nanobody nativeness, because we don't want to transform a single domain from a camelid into a human domain heavy, which has lost its light chain, so it won't be happy. For instance, you don't want, in practice, you don't want to mutate the hydrophilic interface into a hydrophobic one, for instance, and over like properties that the machine learning tools have learned. And then we validated it on, we show that it remained, it isn't detrimental to the binding in some of the strategies. So it remains one of the white type, which is the white type. This is one of our different pipelines. This is the competitive one, but doesn't like check for like nanobody nativeness. And we can see that he lost some of his binding because of that, etc. And it kept like his stability. So this was like my first tool, a native one. I'm designing a native 2 now, trying on a bigger data set and with new transformers that people use in deep learning. And then my second tool in a few minutes is NanoMet. So this is in order to predict the nanobody thermostability. For thermostability, here is defined as the apparent melting temperature when I heat up a sample of my nanobody. I did record it with nano differential scanning fluorimetry, so which is based on intrinsic fluorescence. I did that for over 100 nanobodies that we were able to grab. And based on that, I collated this dataset with dataset available in the literature together to form a dataset of 640 sequences with their melting temperature that were measured from different techniques. So mine's and some in literature were done with nano DSF, but others with just with a dye DSF and with CD spectra, etc. So we combine all this data together in this dataset that makes the biggest dataset of nanobody thermostability so far. That is open source in the literature, and we try to learn from it. It's a small dataset, but we try to learn from it to predict nanobody thermostability by building this pipeline that try to be super robust while learning on the small dataset. So you don't want to overfit, for instance. And what it does is just take your sequences, different representations. So this is the ESM that described like at the beginning of the talk, for instance, a different way to represent your sequence computationally. Then I mix it with different machine learning architectures together, combine them. I did a training in such a way that we make sure that it trains on a diverse type of experimental method, because as I said, it was some of the dataset data points were measured with nano DSF, with CD spectra, etc. So we want to make sure that this won't create any bias into the learning with this cross-validation training, basically. And then we assess it in different ways to make sure that what we learned was meaningful. If our predictions correlates well with the measurement. This was tested on our training dataset during cross-validation, because when you train, basically you save a subset for testing and you see like how it's going, and then you train and you shuffle everything and you train again to have a robust assessment of it. And we were able to show like pretty good correlation with a mean average error of 4 degrees. And yeah, so we can see we're quite good at seeing like very like badly stable nanobodies that were generated for a very specific project. And then like most of them are well predicted. And then we validated it on a external dataset of sequences he has never seen ever, with sequences that are very far away from the training dataset, as we can see here, and some sequences that are quite similar. We can still see like pretty decent correlation. It's obviously like not perfect, but it's the best we can do with the dataset we have. And then how can we apply such a thermostability prediction? So for instance, if you have a library, then you can select the one that actually is the most stable. So what we did, we took a library of the immune repertoires from the native dataset, for instance, and we selected like highly stable nanobodies and poor stability ones. And we show that the one that with poor stability, we didn't even manage to, even though we try hard, to express them, whereas the one with high stability, we managed to express them. And we then measured that melting temperature, and they had temperature very close from the one predicted, like with only like a few degree error. So this is like one way to select. Or if we go back into this in silico pipeline, a generation of nanobodies, if you have like your top 20 or whatever, 100 sequences from your in silico pipeline, then you can select the ones that are actually going to be stable, because maybe some of the ones generated is going to just be the deep learning model will have hallucinated. And so the sequences you're going to see maybe won't even like be stable in its fold. So this is what we try to avoid. Yeah, so this was my different, and again, I want to donate my group. The different tools I've built in my PhD. Thank you for.
No, thank you, Aubin, for taking the time to present to us. I think we have a final question in chat from Jenny. So she was just asking, how high throughput is Nano DSF? Sorry, Jenny, if you wanted to.
So we use the Prometheus machine, which is available at the LMB, and that takes 46 capillaries at the time, 46 or 48, I guess, together. So that is quite handy. Yeah, but then the protein expression and purification was done with GenScript, so I haven't done it.
Nice. Yeah, we've been using just like a plate reader based cypro assay for, but it's pretty crude. I think the Nano DSF is a lot higher resolution. So useful to know.
It is. What we have in our lab is a Tycho. So basically the same thing, but takes 6.
Okay, yeah, less high throughput. Okay, thank you.
Great. Well, if there aren't any more questions, I just want to thank Aubin again for the time you've taken to present to us about your PhD research.
Thank you, Jenny, for your invitation.
Yes, no worries, and good luck with the final push.
Yeah,
and
thank
we'll
you.
hope to hear more about your research in the near future.
Hopefully. Thank you so much.
Thanks, Aubin.
Thank you, guys.
Bye-bye.
Take care now.
Bye, everyone.
Bye.